So our plan was that we have a question time at the end of each session. So if Richard Neher is on the line, we would now continue with Richard. And maybe while he connects and shares his first slide, I will briefly introduce him. So Richard Neher is a professor at the Biozentrum at the University of Basel in Switzerland, who's very active in the field of bacterial and viral evolution and modeling. He's trained as a physicist, and I originally got to know him when he was an independent Max Planck research group leader at our neighboring institute, the Max Planck Institute for Developmental Biology, and talked to him a number of times. He is one of these rare people who really connect a profound knowledge of physics and modeling with a deep understanding of the biology. And I think this puts him in a rather unique position now, and he's been very active in Switzerland and beyond, not only in driving the development of the field, but also in shaping public opinion and keeping us all on track and making sure that we all state our assumptions correctly, etc. So I'm very much looking forward to your presentation, Richard.
Yeah, thank you very much. It's a pleasure to share with you guys what we've been doing. And yeah, so what I was hoping to do over the next few minutes is tell you about two projects. One that has been going on for a long time and now sort of got into the limelight in the context of COVID-19. And then another one which we sort of stumbled into 4 weeks ago and has been keeping us busy quite a lot. Now, as Nuria just told you, we also got completely steamrolled by this outbreak. I sort of remember 8 weeks ago when we were pondering over the numbers in China. It seemed like a very, very remote event that's unfolding there, and we were sort of looking at Singapore whether something happened or not. But this has sort of very profoundly changed, and yeah, sort of we got completely sort of washed up in this between media attention and sort of trying to communicate to the public in some sensible way, doing policy, advising, and sort of doing research and developing tools at the same time has been a real challenge. But so far so good. So the one project that I was going to tell you about is NextStrain, which some of you might have heard about, and that is sort of meant to track the genome and genetic diversity of an outbreak, of a pathogen. And this was initially, so it's an initiative that me and Trevor Bedford, a colleague in Seattle, started about 5 years ago now, mainly to study evolution of influenza. Influenza was sort of our prime target at the time, because this is a virus that sort of continuously circulates and where you need to update vaccines every week, every year or so. And hence it is very important to have an up-to-date picture of the genetic diversity. So when the first genomes of SARS-CoV-2 or HCoV came out in the mid of January in this year, we started to set up a small little NextStrain site for this so that you can see these genomes in the context of other genomes. And by the end of January, this became, or sort of mid-February, this became a huge operation, where suddenly the number of clicks that we had on NextStrain went from a few, about 1,000 a day to 100,000 a day or more. And suddenly we got a bill from Mapbox over a couple of $1,000s, which they nicely waived. But let me just tell you a few things about NextStrain. So if you go on NextStrain right now, I don't know whether this the Zoom thing works well enough to have a sort of full high-res screen, but let me know if you can't see what I'm telling you about. So what you see here is sort of a phylogenetic tree on the left of pretty much all available SARS-CoV-2 genomes to date. This is 2,500, and many of them really just came in the last 2-3 weeks. So the number of genomes that we have to analyze is really growing exponentially. To those that are not accustomed to looking at these kind of phylogenetic trees, what you have here at the very left, that is sort of essentially the genome of viruses that circulated at the very beginning of the outbreak. So the color blue here or purple is China. So in these viruses here, those are going to be many of them. Let me just increase the size of this a little bit. These are going to be the sort of the earliest genomes that that were sampled. And as we now move to the right, we observe that these viruses, they accumulate mutations. So this is just random copying mistakes that the virus replication machinery makes as we go along. So these viruses here, they have sort of one mutation relative to the original Wuhan strain. Then these have two, and so forth. The color here on this graph currently, that is geographic location. So the red ones are from the US, the green, yellow ones are from Europe, and the bluish ones are from Asia. So we can now change this display here slightly to have this x-axis, not number of mutations, but sampling time. And then you get a sort of a picture like this, where sort of these recent genomes here, this is sort of these are genomes from just last week, right? This is sort of March 25, a genome from England. These red ones is sort of a big cluster of genomes from Washington, US. And the early Wuhan genomes, those would be from the beginning of the year or end of last year, where there's a few samples available sort of around Christmas last year from Wuhan. And so the root of the tree, that's sort of the presumed ancestor of this outbreak, that must have been at some point sort of late last year, probably in November. So our current best estimate here is something like late November, early December. And that is sort of only the ancestor of genetic diversity, not necessarily the ancestor of the entire outbreak. Yeah, so then you can sort of, for example, zoom in a little bit, and I prepared this here on a separate graph. So what you're looking at here now is only genomes from Iceland. And the one thing that, for example, that tells you, so Iceland is because they just like last week dumped 300 genomes onto the database. And what is remarkable here is that Iceland at the time had probably like 500 or 800 cases, and they basically sequenced almost every single one of them, but like half of them. And what you see here is that these Icelandic genomes, these are all these dots, they are scattered across the tree. So you have, this clade here, you have lots of these Icelandic genomes, and there's one group of Icelandic genomes here, and sort of a bunch more here, which is basically telling you that these Icelandic genomes are completely, pretty much representative sample of European and US diversity. So this group here could actually be traced back to travel returnees from the US. So they got infected in the US and brought this virus back to Iceland. Most of the other ones had some degree of travel history to Europe, be it sort of skiing holidays, things like that. But then you also observe some outbreaks which sort of within Iceland, where you basically have exclusively Icelandic sequences that group together without any other sequences, suggesting that this was an introduction into Iceland, probably from a tourist that came back from Europe, and then sort of spread locally in little clusters. So that is just to illustrate that these genomes, what they allow you to do is gives these outbreaks structure, right? Sort of without these genomes, Iceland would just be, one number, 800 cases, something like this. But with these genomes, we know that, this is really like 10, 20 different little outbreaks that all got transported to Iceland by airplane, sort of people that got infected abroad, brought it back, and then it initiated local spread. So these genomes, they provide you sort of additional structure to the otherwise completely aggregated numbers that you get from just counting cases or counting dead people and things like that. Yeah, so that was a very brief overview here. Sort of I guess that's sort of most of this I said. So the resolution, the temporal resolution that we have here is about 2, 3, 4 weeks, and that is because we expect to see about one change on the tree every 2 weeks or so. This is quite stochastic. They are over dispersed, so they're not Poisson distributed. So sometimes you observe sort of 3 at once, and sometimes there's none for like 6 weeks. But this is sort of an intrinsic limit to the resolution at which you can differentiate different introductions and transmission chains. But another thing that one has to keep in mind that the overall picture that you get is quite heavily affected by the sampling and sequencing efforts. And Iceland being sort of a very specific example here that sequenced a lot, While if you did the same thing for Iran, you would end up empty-handed, because while they have lots of cases, they didn't so far sequence anything.
Richard, we're getting some comments that you should move closer to the microphone.
Oh, sorry. Yeah, happy to do that. One sort of word of caution, while this might seem like a lot of genomes, it really isn't big data in any sense of the world, because nothing here is sort of independently and identically distributed, right? So total amount of information in 3,000 genomes is probably like 100 bits or so. So there isn't all that. This is not like big data in the classical sense. So just as a word of caution, if one sees a file with 100 megabases or a gigabyte, that does not mean that there is actually. This compresses pretty well. It's pretty small data for all practical purposes. Okay, then the other project that we stumbled into, and that really just started 4 weeks ago, pretty much exactly 4 weeks ago, when a colleague of mine in Sweden called me and told me, well, we really don't know what's coming. Can you give us some idea, some modeling, some prediction, some scenarios, whatever, that could help us to plan hospital capacity? And I told him, yeah, well, we kind of expect to do something, but we'll be operating on sort of very, very sketchy data for the time being. But anyway, this developed into a sort of little web app where you can adjust all sorts of parameters of the classical epidemiological model that's called the SIR model, with a whole bunch of different bells and whistles. Like we have this for different age groups, and there's sort of various different compartments for critically ill, sort of severely ill, and so forth. It's sort of a little bit SIR model on steroids, but basically it's a simple SIR model, a system of coupled ODEs that is solved forward in time. And what it allows you to do is sort of specify a particular population with an age distribution. Age is important because old people tend to be much more severely affected by this than younger people. You can specify how you think different mitigation measures or sort of infection control measures reduce the spread, and then you get sort of an output like this, where you have a model output sort of overlaid with sort of the actual observed cases. So this is data from Germany up until the day before yesterday, where they had 455 deaths. And you see that you can't possibly fit all these curves, because all these the cases and the deaths and so forth, they all have sort of underreporting to various degrees. So we think that in most places, probably the cases are underreported by a factor of 5 to 50. Deaths are probably underreported by a factor of 2-ish. Depends again heavily on the place you look at. And then, of course, any prediction going forward is heavily dependent on what kind of measures are being implemented and how they are complied with. But this is just meant to give you some idea of how, what we know about the virus in terms of severity and epidemiological parameters, and the sort of mitigation measures that were put in place, how they might conspire to generate different future dynamics. So it's quite explicitly not a prediction, but sort of a scenario, in that you make assumptions on what's going on, and then you see how they play out in this model. Yeah, sort of feel free to play around with this. This will going to change quite a bit over the next couple of days. We have sort of a new hosting setup and all sorts of things, but the basic idea and the link right now, which is just neherlab.org/COVID-19, that will continue to work. And with this, yeah, I'm happy to take questions, and yeah, wish you all the best with whatever is being planned.